All right, are there any questions from last time? Any questions? All right, so last time we were discussing the ability of like antibiotics, all right, to stop bacterial infection, right? Because it can get on the bacterial RNA polymerase and stop transcription, right? And we also showed, the principle of survival of the fittest, right, by using an antibiotic. And you see that the mutant bacteria is able to basically take over the population, right? In the case of viruses, you cannot treat them using antibiotics because the virus does not have its own replication machinery. Okay. Now, bacterial cells. So, in this chapter, we are actually looking at genetic variation, right? So, all the different ways in which, in nature, genetic variations are introduced, right? So, bacterial cells can also acquire genes from other bacteria. For example, if you mix a laboratory strain of E. coli, that's the model organism of choice as far as prokaryotic organisms are concerned, that lacks one of the enzymes for making an essential amino acid. An essential amino acid is an amino acid that you have to supply, okay, to the bacteria. That's not something it can synthesize from scratch from other amino acids, right? So you have to supply it in a ready-made form, right? So if you mix a laboratory strain of E. coli that lacks one of the enzymes for making an essential amino acid with another strain that lacks one of the enzymes for making another essential amino acid, right? And if the mixture is allowed to grow together for a few hours, and then you transfer it to a medium that lacks both the amino acids, right? Then normally if there was no gene transfer, right, there would be no bacterial growth, right? Because this one lacks one amino acid, the other one lacks another amino acid, right? It's not able to produce that essential amino acid, and you put it in a medium where both the essential amino acids are missing, right? But if you indeed do that, you see that many rapidly growing bacteria emerge, right, in the new medium. And if you check the genome of the new bacterial strain, it is composed of normal genes for the synthesis of both the essential amino acids, right? So there has been gene transfer between these bacteria. Okay. So that's another way in which genetic variation can happen. Now, genes can be transferred from one bacterium to another by a process which is called bacterial mating. And the ability to initiate mating and gene transfer seen in some bacteria is conferred by genes that are contained in something that is called a bacterial plasmid, right? What are plasmids? Plasmids are small circular double-stranded DNA molecules that are separate from the larger bacterial chromosome, right? And a plasmid that commonly initiates mating in E. coli is what is called the F plasmid or the fertility plasmid. And when a bacterium carrying the F plasmid, which is the donor, encounters a bacterium lacking the F plasmid, which is the recipient, a cytoplasmic bridge is formed between the two cells. And I'll show that to you in a moment in a picture, right? And the F plasmid DNA is replicated, and then it is transferred from the donor through the bridge to the recipient. And finally, the bridge breaks down, and the two bacteria, both of which now contain the F plasmid, can act as donors in subsequent encounters with recipient bacteria. And I think this is best explained in the following diagram, right? So this is the recipient, right, bacteria. It has got its bacterial chromosome. This is the donor cell, the donor bacterium that has the bacterial chromosome, and then it has this plasmid, the circular DNA molecule, right? It's called the fertility plasmid, right? That has all the information that is needed for producing this cytoplasmic bridge, right, between these two guys. And then it's going to make a copy of this, right, and transfer it to the bacterium on the right, right? So the way the copy is made, this is done by what is called rolling circle replication. Because remember, in this circular plasmid, right, the information in the two strands is complementary, right? So each strand, right, the outer circle can serve as a template for the synthesis of the inner strand and vice versa, right? So this thing opens out here, right? The outer strand opens out, and then it starts moving. Once the cytoplasmic bridge has been created by the information that is there in the fertility plasmid, this one opens out, it starts moving to the other cell, right? And simultaneously, right, you start making a copy from the inner template, right, for the cell on the left, okay? Because you have to complete this circle. And here also, as this moves into the other cell, you start using this outer one as a template for synthesizing the inner strand, right? And at the end, what you have is basically you still have the bridge, but you have the fertility plasmid that is still there in the donor cell on the left, and then you have a copy of that in the cell on the right, Okay. As a result, the plasmid DNA has been replicated and transferred to this recipient, which now becomes a donor, right? So it can participate in reactions where it's going to again, transfer the fertility plasmid to some other recipient, right? So this fertility plasmid has got the information that is needed for producing that bridge, right? Bridge in between, right? And then you'll have this DNA replication, which is this kind of DNA replication is referred to as rolling circle replication. So in these pictures, the red DNA is the newly synthesized DNA. The blue one is what you had in the original fertility plasmid. Right? So just keep that in mind. So what I already said before, the F plasmid or the fertility plasmid is necessary for mating because it carries genes that encode some of the proteins that are needed to make the cytoplasmic bridge and also to transfer the DNA. Right? Bacterial mating via the F plasmid does not generate much genetic variation. Why? Because it's not the bacterial chromosome is not getting transferred, right? It's only the plasmid DNA, and it's only a little bit of DNA that is getting transferred, right? So the plasmid DNA contains only a small number of genes. However, occasionally, the fertility plasmid could get integrated into the bacterial chromosome, right? With the result that when it now initiates mating with gene transfer, it can now take parts of the bacterial chromosome with it. So the bacterial plasmid could get integrated into the bacterial chromosome. Then later on, when the plasmid extracts itself, it could take away some of the bacterial DNA, right? Now when the plasmid goes and infects a recipient, right, it could transfer the original bacterial DNA, right? So there could be a lot of genetic variation because of that. Now bacteria can also take up DNA from their surroundings. For example, DNA can be taken up from other dead bacteria in the surroundings. And this process is called transformation, since it can transform one strain of bacteria into another, right? And recall the experiment that we talked about earlier, right? That conclusively demonstrated that DNA is the carrier of genetic information, right? Remember the experiment involving the smooth cells and the rough cells, right? And growing the rough cells in the presence of heat-killed smooth cells, okay? So bacteria can transform, right? All right, any questions so far? Now the most important route by which DNA becomes incorporated into a bacterial genome is what is referred to as homologous recombination, right? Homologous recombination can take place between two DNA molecules of very similar nucleotide sequence, right? So if you have two DNA molecules where the nucleotide sequences are very similar, they're not identical, right, but very similar, then you say that they are homologous, right? So two double-stranded DNA molecules which have regions of very similar homologous DNA sequence, they can align so that their homologous sequences are in register, right? They're like in sync, right? And if you look at the picture on the next page, you can see that, okay? There are two homologous sequences that have come and lined up, right? So the DNA in these regions is very, very similar. So you could have the red DNA crossover, right? On the right, like let's say there's a break here, it crosses over onto the other one, and then so you're left with, two new DNA strands, okay? Where half of it is coming from the one on the left, half from the right. Okay, so the genetic variation can happen in this way, right? Because when similar sequences line up next to each other, there can be a crossover, right? So first, one of them will cross over, then there's some unfolding and all that stuff, that is explained in the book, right? I'm not getting into the details of that. But at the end of the day, the story is that you had this red strand, you had this blue, this green homologous DNA molecule, but then as a result of the crossover, you have partly green and partly red, right, in each of these DNA molecules, right? So this is the figure that I showed you, and the same figure also shows how they can cross over, and the net result is what is called homologous recombination, right? And cells utilize specialized proteins to facilitate this kind of homologous recombination, right? And such recombination enzymes are well characterized in bacteria, but are only beginning to be understood in eukaryotic cells, right? So again, I mean, this business of homologous recombination, it can also introduce a lot of genetic variation, right? Because you don't have the red molecules and the green molecules as before, right? There is some crossover that has taken place, right? So there and there are many possibilities for that. And later on, I will show that this crossover, even in the human genome, right? When a sperm fertilizes an egg, right? There is possibility for a lot of crossovers, okay? That's why no two human beings are identical, right? Even if you're siblings, right? Your source of DNA is the same, it's your parents, right? But then you have completely different characteristics, and even your genome also differs. There are some similarities, but they will not be identical because there is a lot of genetic variation that is introduced by sexual reproduction. All right, any questions?
So the DNA disintegrates like into separate strands, then
Uh,
combines again with something else.
no, there is like what they call a crossover, right? Oh, you're talking about the double-stranded DNA disintegrating?
Yeah, for homologous recombination.
No, it's not going to disintegrate. It's not going to disintegrate, right? I don't have the exact mechanism here, okay, but if you look at the book, right, it'll have the details of that, right? Because see, you have to go basically from here to here, all right? So there are a number of steps involved, right? Because the thing has to uncoil, okay, there'll be some loop and, things like that, but I don't have the pictures here to present that, But there are some details associated with it. But the DNA does not disintegrate, right? It's basically this one breaks up from here, joins that one, and that one is going to join this one, okay? The DNA, and obviously, some steps are skipped here on this slide, right? But the DNA does not disintegrate, okay? The molecules will remain the same, right? But again, I mean, here, see, even if it were to disintegrate, okay, we're not interested in that because here our goal is to show that more variation is going to be introduced, okay? If some of the DNA, let's say, got disintegrated, right? I'm not sure whether it disintegrates or not, but if some of that got disintegrated, that would introduce even more genetic variation, right? You lost some of that stuff, right? That's the message. In this chapter, we are trying to show genetic variation. But if you want to look at the detailed uh, the details of the steps involved, how the thing uncoils and all that stuff, I would recommend you look at the book, right? I assume that it's still there in the newer version of the book. In the earlier version, it was there, So there are a number of pictures over there. And if you make two such exchanges, right? If you make two such exchanges, like one here, then you could replace this red DNA with the green one from on top, right? And the red DNA from the bottom would go on the green one at the top. Okay. So two such exchanges can neatly replace a long stretch of bacterial DNA with a homologous but not identical DNA fragment from another source, right? And the same mechanism can result in I mentioned about the fertility plasmid, the F plasmid getting integrated into the bacterial DNA. So the same mechanism could result in the F plasmid getting integrated into the bacterial chromosome. So here you have the bacterial chromosome, you have the fertility plasmid, right? So you have a break here, right? And this side is linked with this one. The stuff on the left is linked with the other side of this circle. Then you uncoil that, now you got an integrated plasmid. Okay. So from one crossover, from a single crossover, you could integrate the fertility plasmid into the bacterial chromosome. Now later on at some point in time this plasmid might extract itself, right? And in the process it might take some of the bacterial DNA with it. Okay? So this is going to introduce even more genetic variation. Now another mechanism by which genes are transferred is through what are called bacteriophages, right? Or bacterial viruses, right? So viruses invade bacteria just like they invade you and me, right? So bacteriophages are viruses that invade bacterial cells. What is a virus? We've been talking about a virus. I said a virus is not a living object, right? Despite it making you sick and all that, you get the flu and all that stuff. Virus is not a living object. A virus is usually composed of DNA, or it could even be RNA, right? DNA or RNA that is encapsulated inside a protein coat, right? So it has got some DNA inside, it's inside a protein coat. Okay, that's a virus, right? It's not a living organism. It does not have its own replication machinery. It does not have like RNA polymerase, DNA polymerase, ribosomes and so on. Okay, so the reason why we fall sick is when a virus invades the host. Okay, it is going to make use of your cells to make more copies of itself. All right. In the process, your cells will die. Right. Like when you have a runny nose, right? That is the cytoplasm of the dead cells. Right. The cells that have been killed. Right. And you become sick. Right. So a virus enters a bacterial cell and uses the cell's DNA replication, transcription, and translational machinery to produce more copies of its own DNA, right? And its coat protein. Why? Because it wants to make more copies of itself, right? Then the replicated DNA is packaged into additional protein coats to produce progeny viruses which can leave the current bacterial cell and invade other cells, right? Now, when a virus reproduces inside a cell, usually this is lethal for the cell dies, right? So viral reproduction is lethal for is generally lethal for the infected cell, and the cell will burst open, right? And in the biological term for that is lysing, right? So it lyses as a result of the infection, right? And the runny nose, all that stuff is the cytoplasm from the cells that have been lysed, right? Now, when a virus invades a bacterial cell, one of two things can happen, right? The cell could either lyse, right, and a large number of copies of the virus are released, right? So that would happen immediately, right? On the other hand, the viral genome could get integrated into the bacterial chromosome, right? And at each subsequent bacterial cell division, the viral genome is also replicated along with the bacterial genome and passed on, right? So the cell doesn't burst, but every time the cell divides, you're getting more copies of the bacteria of the viral information getting reproduced, right? Now, an environmental insult such as exposure to ultraviolet radiation can induce the viral genome to leave the host chromosome and begin a lytic phase of viral replication, right? So most of the time, route 1 above is followed, but on rare occasions, route 2 is also followed, right? Like you may have heard that HIV virus can be dormant and all that for years, right? That's what happens. Of course, HIV is a retrovirus, and I will talk about that in a moment. You know, it's not carrying DNA, it's carrying RNA, right? So it's a little bit different, right? But again, I mean, it's staying dormant because it's integrated into the host chromosome, right? And then it'll stay for years, and then when it exists, that's when the activity starts again. So the viral genome usually integrates with the bacterial genome by what is called site-specific recombination, which is carried out by an enzyme called integrase. It is not homologous recombination, right? It's something that's called site-specific recombination. Now, on leaving a host chromosome, the viral DNA will occasionally remove itself inaccurately and bring along the neighboring piece of host DNA in place of part of its own DNA, right? So then again, more genetic variation, right? So this host DNA will be packaged into a virus particle along with the viral DNA. So when this new virus that is carrying part of the host DNA infects a new host, it introduces along with its viral DNA that is derived from the previous host, right? So that means there's going to be a lot of genetic variation, right? Because that bacterial DNA, which normally shouldn't have got into the host DNA, is going to get in, right? So this bacterial DNA can become part of the new host chromosome in at least a couple of ways. The incoming virus integrates into the new host chromosome, or if the incoming virus does not destroy the host, the passenger bacterial DNA can become a permanent part of the host genome by homologous recombination, right? So lots of possibilities. And this process is called transduction. All right, any questions so far? Again, I mean, the take-home message in this chapter is basically just to show the large number of possibilities, right, by which genetic variation can arise in nature, right? And then we'll see how we can do it in the lab, right? In nature, it will arise just by chance, okay? It's a random thing. It may happen, may not happen. But in the lab, we can do it pretty precisely, right? And that's what the next chapter is going to be about. Now, another source of variation in bacteria and also in eukaryotic cells are what are called transposons. So many bacterial and eukaryotic genomes contain stretches of DNA which are called transposable elements or transposons, which can move from place to place within the chromosome by a process that is called transposition, right? Transposons move within the DNA of their host by means of special recombination enzymes, which are called transposases, encoded by the transposable element to create great genetic diversity, right? And let's look at the picture, right? So there are two possibilities. So let's say this is the donor DNA, right? And this is the target DNA, right? So there are two possibilities. In non-replicative transposition, from the donor, this thing will be cut out and pasted on the target, right? So you get a new DNA sequence, right? Because of some enzymes that can do it. If you have replicative transposition, then this information is not lost, okay? So the donor doesn't lose anything, but a copy is made of the donor information and pasted on the recipient, right? So there are enzymes, right? And there are sequences which can effectively move around in the genome through this mechanism of transposition, right? So as I showed in the picture, in non-replicative transposition, The stretch of DNA from the donor is removed and incorporated into the recipient, while in replicative transposition, the donor DNA remains intact while a copy is made for incorporation into the recipient DNA. That's exactly what I showed in the figure, all right? And let's make the observation that most alterations in the genome are harmful to the individual bacterium, right? And if you make too many alterations, right, you're probably not functional anymore, right? And so the bacteria with these kind of alterations are quickly eliminated from the population. However, alterations also confer survivability in a changing environment, right? As was demonstrated by the example involving the antibiotic rifampicin, right? Rifampicin. I discussed that last time. That if you treat with that antibiotic, most of the bacteria are dead, right? Some of them survive, right? And those are the ones where the genome has been altered, right? They are able to survive. All right, so we have already discussed the source of genetic change in the case of bacteria, right? We talked about the fertility plasmid, then about transposable elements and so on. Okay, then bacteria getting transformed in the presence of other dead bacteria, right? So there are many different sources. Now we are going to look at sources of genetic variation in eukaryotic genomes, right? Like including the human genome, right? So unlike bacterial DNA, eukaryotic DNA has got both coding and non-coding regions, right? What are the coding regions called? Exons. And the non-coding regions? Introns. Good. Yeah. So in prokaryotes, in the case of bacteria, the rate of cell division is very high, and hence there is strong selective pressure to minimize the amount of superfluous DNA in the genome. All right, and maybe under that pressure, bacteria basically got rid of their introns. Right. Now, eukaryotic genomes are also characterized by a large amount of gene duplication, right? You have many copies of the same gene. Like if you look at the beta globin gene, we have 5 copies of that gene, right? And different copies are more suitable at different stages in life, right? So in eukaryotic genomes, there can be several genes belonging to the same gene family, and the most well-documented example of that is the beta globin gene family, right? And the human genome has got a total of 5 beta globin genes, right? And these genes encode the beta subunits of the various hemoglobins produced at different times during embryonic, fetal, and adult life, right? So so hemoglobin is made of how many subunits? Anybody? No. 4 subunits. So you're using only even numbers, huh? You don't want to try 3. There are 4 subunits, 2 alpha subunits and 2 beta subunits, all right? All right, any questions? So we will now see how that gene duplication takes place, right? How is that possible, right? So let's look at again, I mean, explaining with the picture is much easier, all right? So suppose you have the globin gene, all right? This is, one double-stranded DNA, right? This is another double-stranded DNA, and this is where the globin gene resides, okay? So normally, if there is going to be a crossover, right, the 2 homologous sequences, they'll line up next to each other, and there'll be a crossover, right? And you'll get 2 functional globin genes, all right? Now, it may happen, right, that there's some sequence similarity between this end, right, and that end here, because of which the alignment is like this. This is improper alignment, right? Now, if there is a crossover, right, see what happens, right? One of the chromosomes, right, the long one, it has got 2 copies of the globin gene, right? The other one has nothing, right? It has the short chromosome. So if there is a cell that inherits this short chromosome, that has no information, that cell will be eliminated from the population. The one which has 2 copies, right, that will survive, right? And this procedure can be repeated, at least in the human genome. There are 5 copies of the beta globin gene, right? So this is how gene duplication can occur, right, in eukaryotic cells, right? In the same way, the duplication might also apply just to exons. The entire gene might not get duplicated, right? Remember, you told me that for eukaryotic genes you have exons and introns, okay? So another possibility is that the exon duplication could occur, right? And that's in the next slide, right? So you have exon A, exon B, right, over here, and in a normal crossover, they would align up properly, right? And the crossover will take place, you'd get a functional gene. If there's an improper or unequal crossover, maybe this one aligns up in this improper fashion. They have the same information, exon A, exon B, Well, again, homologous chromosomes, all right, not identical, right? So very similar information. And then you have a crossover. As a result of this crossover, what happens is that the long chromosome now has exon A, exon B, and exon B again. Okay, two copies of exon B. The short chromosome has only exon A, right? Now, one more important thing to note here is that, remember, for eukaryotic genomes, all right, or eukaryotic genes, you have coding and you have non-coding regions, all right? So if there is a crossover in the non-coding region, right, even if something got messed up, even if some nucleotides got lost, like you were saying some of the DNA might be, nothing is going to happen. You will still have a functional gene, right? So that's why the presence of introns, right, in eukaryotic genomes, right, greatly increases the probability that this kind of crossover, right, will still result in a functional gene, right? And I'm giving you a protein that is functional, right? So this actually, if you had only coding regions, or if there's a crossover, if some mistake is made over there, then you're finished, okay? That will not, the protein produced is not going to be functional, right? So this is also another source of, genetic diversity, right? In the case of eukaryotes, the fact that you have introns means there are lots of places where crossovers can take place, right? And the function would not be changed, but the genetic information has been changed, right? The functional protein would not have been changed. All right, so let's just run through the text. It's essentially the same information that I showed you in those two pictures. So gene duplication is thought to occur from a rare recombination event between two homologous chromosomes. Consider the case of the beta globin gene duplication. Instead of aligning properly for a crossover, which is shown at the top of that picture, right? The proper alignment for a crossover is at the top, right? It aligns like shown in the middle, right? So the two chromosomes align in an improper fashion as shown in the middle of the figure. After the crossover, the long chromosome has got two copies of the globin gene, while the short chromosome lacks the original globin gene. And consequently, the individuals that inherit the short chromosome, they are expected to be eliminated from the population because they don't have a functional globin gene, right? So no functional hemoglobin, right? While the individuals that inherit the long chromosome, they would be expected to have two beta globin genes instead of one, right? And we all have like 5 copies of that. So it has happened at some point in evolutionary time, right? This kind of gene duplication. Now genes encoding new proteins can also be created by the recombination of exons, right? And the general scheme, which is shown in the subsequent figure that I showed you, right, is the same as before, except that an exon within a gene, rather than the entire gene, is duplicated, right? The exon got duplicated, not the entire gene. And this also greatly increases the chances of genetic variation in functional genetic variation or genetic variation that preserves function in eukaryotic cells. So without introns, this is the point that I made, without introns there would be very few sites on the original gene at which a recombination exchange between homologous chromosomes could duplicate the domain without damaging it, right? The important thing here is without damaging it. That gets back to your point, okay? So therefore introns greatly increase the probability that DNA duplications will give rise to functional genes encoding functional proteins. Moreover, the presence of introns greatly increases the probability that a chance recombination event can generate a functional hybrid gene, right, by joining together two initially separate exons coding for quite different protein domains, right? So a lot of possibilities of variation, and this is referred to as exon shuffling, okay? Just like you shuffle a deck of cards, right? This is like exon shuffling. Now, as we've said, in the case of the bacterial genome, right, you have transposable elements, all right, that can move about freely in the genome, right, with the help of these transposases. In the case of eukaryotes also, you do have transposable sequences. In fact, about 10% of the human genome consists of two families of transposable sequences. Transposable DNA elements move from place to place by the mechanisms that have been discussed earlier for prokaryotic transposons. All right, I talked about replicative transposition and non-replicative, right? Gene transfer I showed you in those two diagrams. Same thing happens even in the case of eukaryotes. However, for eukaryotes, there are also what are called retrotransposons, for which, retro means it's against the normal flow of genetic information, right? The normal flow of genetic information is from DNA to what? RNA and then to protein, right? So if you go from RNA to DNA, right, that is going to be retro. The HIV virus is a retrovirus because it normally carries RNA and it has to use an enzyme, all right, that is called reverse transcriptase, right? So you have transcription, right? The reverse of transcription is carried out by an enzyme that's called reverse transcriptase. So that RNA is going to be copied into a DNA, right? So you have a retrovirus and you have reverse transcription going on. And I'll explain this in more detail a little bit later, right? So for eukaryotes, there are also retrotransposons for which an RNA copy is first made using RNA polymerase, following which DNA copies are made using reverse transcriptase. Now notice what is going to happen in this step, right? Why would a cell do that? Okay, because see, when you copy, in the case of eukaryotes, okay, when you copy the DNA, right, into RNA, right, when you get messenger RNA, what happens? Is the information in the messenger RNA exactly the same as what you copied from the DNA? What? Well, it's complementary then, and then after that what happens? In eukaryotic messenger RNA, what happens? Are the introns copied into the messenger RNA? No, they're taken out, right? They're splicing, all right? So that means if you go from DNA to RNA, right, you've gotten rid of all the non-coding information, right? And now if from that RNA you synthesize the DNA, that's a lot more information to carry. Only the real required information is carried along, okay? Because in nature, usually, things don't just happen redundantly, okay? There is a purpose for it. So they're trying to optimize, not waste resources, all right? So transposition by reverse transcription is shown on the next page, right? So here you have the donor DNA, right? You have RNA polymerase that will act on the donor DNA to produce RNA. This RNA, if you're looking at a eukaryotic cell for messenger RNAs, all the introns are removed, right? All that unnecessary baggage is gone. Now reverse transcription is carried out using the enzyme reverse transcriptase that gives you DNA without all of this additional intron information, right? And now this can be copied onto a target DNA, right? The information that really needs to be copied, right? So you do have retrotransposons which are able to actually do this. So examples of human retrotransposons are the so-called L1 transposable element and the ALU sequence. Again, I mean, for us engineers, this is just like Greek. It's just some terminology. Okay, we are not going to go into the details of that. Like, if you want to, you can Google it up and get more information and all that, right? But you don't have to, right? Now, the evolution of genomes has been greatly accelerated by the presence of these transposable elements. For example, the insertion of a transposable element in a regulatory region for a gene, right? What is the name of the regulatory region for a gene? The primary regulatory region where transcription starts, what is it called? Anybody here? What? Promoter, right? Right? I mean, there can be others too. Yeah, this is not the promoter for the flats and all that, okay? This is a different promoter, right? So the evolution of genomes has been greatly accelerated by transposable elements. The insertion of a transposable element in a regulatory region for a gene will often, by disrupting or adding short regulatory sequences, it can have a dramatic effect on gene expression, right? Because we have seen that there has to be that Datta box, right, and some other stuff that the RNA polymerase has to see before it will do the transcription. So if there is something in there that goes and changes stuff, right, that moves around, right, that can dramatically alter gene expression. All right, any questions? Now I'm going to talk a little bit about what are called retroviruses. Okay, I said retro means it's reversing the normal genetic information, flow of genetic information. So another source of genetic variation in eukaryotes is the activity of viruses, right? Like bacteriophages. Bacteriophages are viruses that attack bacterial cells, right? So like bacteriophages, viruses that infect eukaryotic cells are fully mobile elements that can move into or out of the cells. All right, we've seen like for bacteria, the virus can come in, right? The viral genome can get integrated into the host genome, then make more copies of itself and so on. Okay. So now viral genomes, as I said before, can be made either of DNA or of RNA, and they can be single-stranded or double-stranded. If it is an RNA genome, it will probably be single-stranded, right? RNA usually occurs in nature as a single-stranded molecule. If it's a DNA genome, it will be double-stranded. Now an important class of viruses is what are called retroviruses, right? Which reverse the normal flow of genetic information, right? And the virus that causes AIDS, HIV, is a retrovirus, right? So these viruses will have a genome which is made up of RNA, and they have a protein code that encapsulates the genome, and they also carry the enzyme reverse transcriptase, right? Because you need reverse transcriptase to copy the RNA into DNA, right, before integrating it into the host genome, right? So the schematic diagram for a retrovirus hijacking a host cell is shown in the figure, and I'm going to try to explain that, right? So here you have a host cell, right? This cell is going to be invaded by this virus that has got this blue stuff inside, that is RNA, the RNA of the virus. That green dot over there, that is the enzyme reverse transcriptase, right? Then you have the coat around it, so there is some lipid around it, and there is a protein coat, right? These Y's over here, that's the protein coat of the virus, okay? Now this virus is going to enter this cell, okay? How? It is going to get rid of its protein coat, right? And then stuff all of its contents inside, okay? Like including the reverse transcriptase and this RNA, right? So what is the first step? It has got RNA, not DNA, right? So using reverse transcriptase from this RNA, you can produce a complementary DNA, right? So the RNA is shown in blue, the DNA is complementary DNA is shown in red, okay? Now using this as a template, right? You can produce double-stranded DNA. So now you have the viral DNA here. It started out as RNA, goes through all these steps inside the cell. Now you have the viral DNA. That can now get integrated into the host DNA, right? And so what is the purpose? So now once it's integrated into the host DNA, the cell has its DNA replication machinery, transcription, translation, it can do everything. So now this is going to get transcribed. There's going to be transcription. So it's producing, so that transcription, what is it going to produce? It's going to produce, first of all, the RNA of the virus, that's going to get produced, right? Then it is also going to produce the information that the messenger RNA that is needed to produce reverse transcriptase, right? Because see, the goal of the virus is to make more copies of itself, right? Which means that at the end of the day, it has to have the protein code, more RNA, more reverse transcriptase, right? So there's going to be transcription to produce more copies of its RNA. Then some of the other RNAs which are coded in here, they'll produce the viral proteins, right? Transcribed and then translated into viral proteins, and also produce the enzyme that is reverse transcriptase, because that's also a protein, right? So it's going to get first transcribed into messenger RNA, then translated into protein. After that, this is now going to be packaged into new viruses, right? Each green dot, right? This blue stuff here, right? And then it's going to be packaged inside the protein code, right? Then if the cell dies, it's going to release a lot of copies of this, right? Okay. So that's how the retrovirus actually infects a host cell. Okay, that's how the AIDS virus gets in. Okay, the HIV virus, right, that causes AIDS, that'll get in that way and make many copies of itself. And it can lie dormant for a while. Like, I mean, if the cell doesn't die immediately, maybe more copies are being made, or it may not right away start making more copies of itself and make the cell burst. Every time the cell divides, okay, this viral genome is copied, all right? And then, in response to some insult, there'll be a, basically a massive production of viruses. Okay. It can lie dormant for some time, but remember, every time the cell is dividing, copies of the virus, the genomic information is getting copied, right? Okay. So any questions so far? So this just explains what I showed you in the figure. The enzyme reverse transcriptase is first used to make a single-stranded complementary DNA copy of the viral genome. The enzyme DNA polymerase, which is present in the host cell, okay, the cell has its own replication machinery, is then used to create a double-stranded DNA copy of the viral genome, which is then integrated into the host DNA, right? And transcription and translation from the integrated genome will produce copies of the viral RNA, reverse transcriptase, and the code protein, and all of those can be packaged together to produce additional retroviruses. Okay. Now, retroviruses which have picked up host genes, they can make cells cancerous, right? Very few human cancers. There aren't too many human cancers that are caused by viruses, right? So Like ovarian cancer is one of them for which they give shots and all that, right? The papillo something virus, all right? That's one of them that can cause cancer, all right? But most cancers are not caused by viruses, all right? But in the case of animals, viruses can be a prominent cause of cancers, right? And one example of that, or the most well-known example of that, of a cancer-causing virus is what is called the Rouse sarcoma virus. Right? Sarcoma is a type of tumor, right? Like if you talk about cancers, you can have carcinoma, all right, adenoma, and so on, right? Sarcoma, lymphoma, and so on. Okay, sarcoma is a kind of tumor, right? Cancerous tumor. So the most well-known example of a cancer-causing virus is what is called the Rouse sarcoma virus. This virus can pick up a gene from its chicken host by the mechanisms that we have talked about, okay? How the virus can pick up genes, all right? So this gene, which is called SRC, is unnecessary baggage from the point of view of the virus. Remember, we showed about a virus getting into a bacterial cell. Okay, when it's leaving, it can take away part of the bacterial genome, all right? So here a virus, let's say, has infected a chicken cell, all right? When it's leaving, it takes away part of the chicken genome, right? And it could pick up a gene that is involved in the control of cell division, right? So this gene, which is called SRC, is unnecessary baggage from the point of view of the virus, but it has got profound consequences for cells that are infected by the virus. The normal SRC gene in the chicken genome encodes a protein kinase that is involved in the control of cell division, right? What is a protein kinase? Anybody? Not remove, it adds. Phosphatase removes, right? Now, if you have a protein kinase, okay, if it adds a phosphate group, right, to some protein, okay, what is going to happen to the protein? When you add a phosphate group, right, you're adding negative charges, right, two negative charges, which means that the three-dimensional conformation of the protein will change, okay? And in a lot of situations, these kinases, they are able to turn proteins on or off, right? Either active or inactive, right? And the cell division in our bodies is basically controlled through protein kinases and phosphatases, right? So, so this SRC gene actually encodes a protein kinase, right, which is involved in the control of cell division, right? And it is called a proto-oncogene. If it's a cancer-causing gene, it becomes an oncogene. Proto-oncogene is before it causes cancer. I'm going to be discussing cancer also in detail in a subsequent lecture, right? But for now, I'm just letting you know that the problem is that, you do need genes, all right? You do need genes to turn on cell division, because if you have a wound or a cut, that needs to heal, right? So you need accelerated cell division, but it should be only in response to the stimulus. In the case of cancer, the genes are turned on. Okay, so it's a proto-oncogene. So it's a good gene that turns on cell division. Okay, before it becomes an oncogene, right? So the SRC actually codes for this proto-oncogene. However, the version that the virus is carrying, right? Because when it pulls it away, it's probably not doing it accurately, right? From the chicken host, the gene carried by the virus is not quite identical to the normal cellular gene. And the difference gives rise to its ability to cause a cancer, right? It can cause the chicken cell to multiply uncontrollably, right? Resulting in a tumor and cancer. So this mutated SRC gene is called an oncogene, and it causes the infected cell to divide uncontrollably, right? Resulting in a tumor, and finally it becomes an aggressive tumor that's able to invade other territory. Then it becomes what is called malignant, and it's a cancer. But again, there is a chapter that is devoted to cancer. In the beginning, I was thinking, because I focus only on plant genomics, I won't cover that. But I think I'm going quite fast, okay? And you guys are keeping up pace. It looks like, studying, and at least you're able to answer my questions. So I will be able to do that chapter too, right? So, all right. So now we come to the last topic in this chapter, right? Which is which basically focuses on showing the wide amount of genetic variation that arises because of sexual reproduction in eukaryotes, right? Now, in the case of bacteria, they will reproduce asexually, and this gives rise to offspring that are identical to the parent, right? Sexual reproduction, on the other hand, which is what you have in the case of eukaryotes, it involves the mixing of genomes from two individuals to produce offspring that are genetically distinct from one another and also from both their parents, right? Sexual reproduction occurs in diploid organisms. Like we are diploid, we have two copies of each chromosome, right? So sexual reproduction occurs in organisms like that have 2 copies of each chromosome. Bacteria have 1 copy of each chromosome. They're haploid organisms. So they divide by a fission type of mechanism, right? Whereas diploid organisms, their cells divide differently, right? I mean, at least their reproductive cells, right, divide in a different way. Okay? That introduces a lot of genetic variation. So sexual reproduction occurs in diploid organisms in which each cell contains 2 sets of chromosomes, one inherited from each parent. The actual cells that carry out sexual reproduction in diploid organisms are called the germ cells or the gametes, right? These gametes, which are haploid cells, because see, when an egg and a sperm fuse, right, you have to have the correct number of chromosomes, right? So if you don't have a reduction in the number of chromosomes, right, going from the diploid cell to the egg or the sperm, then there will be problems, right? And you do see those kind of problems. Like for example, many of you might be familiar with people with Down syndrome, right? In Down syndrome, what happens is that the total number of chromosomes is not 23 pairs. Okay, one of the chromosomes, like number 21, right? Instead of having 2 copies, they got 3 copies. Okay, because something failed during this process of production of the sperm and egg, right? So, I mean, most cases where there are mistakes like that are made, people don't survive. They don't make it. Okay, so they will die in utero or in the womb, all right? But if they make it, they will have abnormalities. Okay, and we'll talk about these things a little bit later. So here in this chapter, the focus is just on showing the genetic variation. Now the haploid germ cells are generated from their diploid precursors by a special type of cell division which is called meiosis, right? And we will cover meiosis in detail in a later chapter, both mitosis and meiosis, right? Mitosis is just the division of the somatic cells, okay, how, our organs and all that grow, right? And meiosis is the mechanism by which the egg and the sperm are produced from their diploid precursor cells, right? So let me just straight jump to the picture, right, and start explaining, then I'll go over the text. So here, see, if you're looking at a human cell, there are 23 pairs of chromosomes, right? A paternal copy and a maternal copy for each one of them, right? Now we will focus attention only on one chromosome, right, and see, what changes, okay, or I mean, how do you end up getting the egg or the sperm, right, containing a copy of each of these chromosomes, right? So these two are homologous chromosomes, right? One is the paternal copy, right, the one that is marked, and the unmarked one or the blank one, that is the maternal copy of that chromosome, right? Now, normally, you have a diploid cell with 2 copies of that chromosome, right? You want a haploid cell, right? You would want like 1 copy of each chromosome, right? So the easiest thing to do, right? Easiest thing to do would be for this cell to divide in a fission type of fashion, right? Where the chromosome on the top goes to one daughter cell, the chromosome on the bottom goes to the other, right? And that's the end of the story. So you have the haploid, egg and sperm, right, to form a new organism, right? But that's not how it happens, right? For some unknown reason, right? Initially, so you don't have just one round of cell division without DNA replication, which could produce your haploid gametes, right? For some unknown reason, that doesn't happen. Instead, there is one round of DNA replication. So the paternal copy is replicated, maternal copy is also replicated, okay? And then to get haploid gametes from here, you're going to need 2 rounds of cell division, right? I mean, because each gamete should only have 1 copy of this chromosome, right? You will need 2 rounds of cell division. Okay, that's how it happens, all right? But before that, remember the paternal and the maternal copies are actually homologous chromosomes, right? I mean, because if you're looking, say for example, the gene that is coding for hemoglobin, right? The version you inherited from your father is very similar to the one that you inherited from your mother, and hopefully both of them are functional. So those are homologous chromosomes where the DNA sequence is similar but not identical. So once the DNA replication has taken place, in meiosis, what happens is that the homologous chromosomes, they will line up next to each other, right? They are homologous chromosomes. Guess what is going to happen next? There is going to be crossover, right? So a piece of this one will go to the other one and vice versa. Okay. So on an average, there is at least one crossover on each chromosome. There might be many more. Okay. And then you are going to have 2 rounds of cell splitting, right? 2 rounds of cell division. So first round, the stuff on the left is here, on the right is here. Then one more round, then you get 4 different gametes. Okay. So that's what. So this is called meiosis one, the first cell division of meiosis, and this is called meiosis two. All right. Now look at the amount of genetic variation that has been introduced. All right. So if you step back and think about it, if there was no crossover, right? So suppose there was no crossover here, you just had DNA replication, right? And then you produce 4 gametes from here. Okay. How many different types of gametes would you have? First, let's say there's no crossover. All right. No crossover. Just looking at this, okay? Now from here I'm going to get 4 different gametes, okay, each containing 1 copy. So how many different types are you going to have? 2, right? Now because of crossover, how many are you going to have? If you have 1 crossover on this chromosome? 4, right? Because of if there's only 1 crossover, maybe there might be many more, okay? So just for 1 chromosome, right? When you're going from the diploid cell to this guy, to these haploid gametes, okay, you are having 4 possibilities, right? If you have 23 chromosomes, how many possibilities are you going to have? You're all engineering people, okay, so I'm asking you, yeah. If you have 23, so for 1 chromosome, there are 4 possibilities, okay? And let's say these are independent, okay, so how many possibilities are you going to have when you have 23 chromosomes? Yeah, go ahead. 4 to the? 4 to the power of 23, right? Can you imagine how large a number that is? Okay, and this is just the egg, right? Because the egg and the sperm have to fuse. Okay, so there are 4 to the power of 20. Actually, the number is more if there is more, if there are more crossovers. Okay, 4 to the power of 23 possible distinct eggs that are being produced. Okay, similarly, 4 to the power of 23 distinct sperm that are being produced. Right. Then if you combine them together, it's 4 to the power of 23 multiplied by 4 to the power of 23. All right. So you can imagine how big a number that is. Right. So that's why it's not surprising that no 2 individuals, right, have the same genome, right? You might have the same parents, doesn't matter. Unless you're identical twins, you're not going to have the same genome. Okay, so I'll just go over the text explaining this stuff. And meiosis we'll cover in detail later. Here I just want to show the large amount of genetic variation that occurs because of meiosis. So our main focus here is on showing the staggering amount of genetic variation that results from meiosis. A schematic diagram showing how meiosis produces haploid gametes from a diploid precursor cell is shown in the figure. For clarity of presentation, we are focusing attention on only 1 pair of homologous chromosomes. And during meiosis, the chromosomes that first duplicate and during meiosis, the chromosomes first duplicate and then the homologous paternal and maternal chromosomes line up next to each other, and next crossovers take place by homologous recombination, and 2 rounds of cell splitting occur to produce the 4 different haploid gametes. So clearly, in the absence of a crossover, each pair of homologous chromosomes will give rise to 2 different types of gametes, depending on whether it contains the paternal homologue or the maternal homologue. And with 23 chromosome pairs in the human genome, each individual can produce 2 to the power of 23 possible gametes. The actual number of possible gametes is much larger because of crossovers. On an average, you'll have at least 1 crossover on each chromosome. To develop a new organism, 2 such gametes of the opposite kind, egg and sperm, will have to fuse to form the single diploid cell that will undergo repeated mitosis to develop into the new organism. Right? Thus, the number of possible ways in which the genetic material gets reshuffled during sexual reproduction is just staggering. Right? It's mind-boggling. This is the reason that it is not at all surprising that no 2 individuals except identical twins have the same genome. All right, any questions? So we are done with this chapter on genetic variation. Now we'll move on to the next chapter, which is DNA technology, where we are going to introduce, all right, artificially introduce precise genetic variations in the lab. Okay, we can do that today. All right. Before that, any questions on genetic variation? Again, I mean, like, see, as an engineer, or in terms of applications of engineering in genomics, you would not worry too much about genetic variation because that's what has happened over evolutionary time. That's not what happens unless you're a bacterium. That's not what happens over 1 day, 1 month, or 1 year. Okay, it takes a much longer time for those variations to take place. Right. But this one is going to be important. The stuff that we're going to do in the lab, and it'll make you familiar with many of the procedures that are used in the lab to do genetic variation, or to bring about changes in the genome of an organism. So in this chapter, we will look at some of the modern technologies that enable us to create genetic variation in the laboratory setting. Humans have been unknowingly experimenting with DNA. For example, the crossbreeding between plants. Okay, we know all this grafting between different plants, right? And between animals, they are really instances of gene shuffling, right, or genetic shuffling. Even there is a famous pea experiment that Gregor Mendel did in the early 1900s. Okay, like if you have peas, you want to grow, tall plants. Okay, with certain characteristics. Okay, how do you breed them, right? Of course, he didn't know anything about genes and all that, but he was pretty smart. He figured out a way to do that, to bring about the phenotypic change, right? Now, in the early 1970s, it became possible for the first time to isolate a given piece of DNA out of the many millions of nucleotide pairs in a typical chromosome, right? And this in turn made it possible to create new DNA molecules in the test tube and to introduce them back into living organisms. And this is referred to as recombinant DNA technology or genetic engineering. Right? Today we can do that. Right? Now, there are a number of techniques that have made this kind of technology possible, right? So, one of the techniques is the discovery of what are called restriction endonucleases, right? So these restriction endonucleases, these are enzymes that will cut DNA at certain specific nucleotide sequences, right? So there are different enzymes. So if you have a particular nucleotide sequence, like say for example, if you have this restriction nucleotide HAE3, right? Then if you have the sequence GGCC, then it is going to basically cut between GG and CC, right? On the other hand, if you look at ECO-RI, right? Then if you have this nucleotide sequence GAATT, right, TTC, it is going to cut it in this fashion, right? So at very precise locations, it's not like, you know, I covered one of those methods where you can pass like maybe DNA or protein, through a plunger and use mechanical shear. That is going to cut things in an unpredictable fashion. Okay, it's going to vary from one run of the experiment to the other. Here, the restriction nuclease is going to cut things in a very predictable fashion. I'm not asking you to memorize all these, okay, where it cuts. You're not a biologist, you're not doing those experiments, okay, but just keep in mind that this is an important development, okay, that allows us to cut DNA in a very, very precise fashion. And there are others too, okay, there are just two of them listed here, then there are more on the next page. Another one, ALU1 is going to cut up in this fashion. NOT1 will cut up in this fashion. HIN3 will cut up in this fashion, right? So the point to note is that restriction nucleases cleave or cut DNA in a very predictable and replicable manner. Unlike mechanical shear, which will fragment DNA, but there is no way to predict where the fragmentation will occur. And furthermore, the fragmentation locations due to mechanical shear will change from one run of the experiment to the other. With restriction nucleases, that's not going to happen, right? If it sees that sequence, it'll cut it and give you those pieces, right? So it's like given a large amount of DNA, you can cut it up into pieces, all right? And depending on the sequence, the pieces might have some pattern, okay, that you might be able to study. Right? So the first item is endonucleases, restriction endonucleases. The second item, right, the second advance is what is referred to as gel electrophoresis, right? And your doublies here, so you will love this, okay? In gel electrophoresis, what are you doing? Remember, we already discussed, all right? What kind of electrophoresis did we discuss earlier? Or where did we discuss that? Does anybody remember? We talked about electrophoresis before. Not with DNA, but with something else. With what? Not plasma membrane. What were we trying to separate? No, we want to separate proteins, okay, on the basis of their weight, right, molecular weight, and on their charge, right. So on the basis of their size, all right, or weight, right, the method we used was called SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis, right, where we try to separate proteins based on their size. And then isoelectric focusing was used to separate proteins based on their charge. Right? But the principle was the same. You basically had a slab here, right? And then you coated the protein with something negatively charged. Then that thing migrated depending on the electric field, right? Here, see, if you look at DNA, okay, DNA has got a sugar phosphate backbone. So it already has what kind of charges? Negative charges. It's already negatively charged. So you place it in an electric field, right? It is going to move, right? and the amount by which it will move will depend on the length of the DNA, right? Right, the smaller ones will move faster, right? So here, so you can separate out the DNA based on its size, right? So you have double-stranded DNA. You cut it. Let's say you cut it with a number of restriction endonucleases. So you get smaller DNA pieces. Same way you get some small DNA pieces. Then you run this, okay? You run gel electrophoresis on it. So you have a slab of agarose gel, right? And you have an electric field where the top is negative, this one is positive, right? So this is negatively charged, so it will have a tendency to move, right? And then if you run this for some time, it will form the bands. Depending on the size, it will move to different locations, right? So this is the technique that is used to separate out DNA fragments on the basis of their size, right? And the thing is, if you cut it with, if you have a, let's say a strand of DNA, right, you do not know what the sequence is, but you've cut it with different nucleases, right? Then, with nuclease number 1, you got some pattern here. Nuclease number 2, you got some pattern here. You might be able to compare this with some patterns that you get with, other known DNA sequences and get some idea, right? You still won't get the complete sequence, right? But at least it will be better than knowing nothing at all, okay? So that's what is used. So in gel electrophoresis, which is what I showed you in that picture, any questions on that? So in that picture, fragments of DNA are loaded onto a slab of agarose gel and they are subjected to an electric field as shown in the figure. Since DNA is negatively charged because of the phosphate in there, right? The fragments will migrate towards the bottom and the distance that each fragment travels will be inversely proportional to its size, right? So if one incorporates the radioactive 32P isotope, right? This is the radioactive isotope of phosphorus. Remember, in DNA, you have sugar, you have a phosphate, right? For that phosphate, if you put in the radioactive phosphorus, right, I'll be able to track the location of that DNA molecule, right, by a technique called autoradiography, right? I might be able to capture that information on a photographic plate and so on, okay? So if one incorporates the radioactive 32P isotope of phosphorus in DNA before electrophoresis, then the DNA fragment positions after electrophoresis can be detected by using the technique of autoradiography, right? And by comparing the sizes of the DNA fragments produced from a particular region of DNA after treatment with different combinations of restriction nucleases. So you'll be running, for each restriction nuclease, you'll be running a different electrophoresis, right? And then you look at those locations. So with that information, a physical map of the region can be constructed showing the location of each cutting site. Such a map is known as a restriction map, right? But again, it's not very useful. Today there are much better techniques. So in fact, you can go and actually sequence the entire DNA, right? Find out all those letters, A, G, C, T, right? So a restriction map, however, does not provide the complete sequence information for the entire DNA sequence. To determine the complete DNA sequence of a given DNA strand, one can run a 4-lane gel electrophoresis, which I will explain in a few minutes, following 4 separate DNA synthesis reactions as shown in the figure, right? And let's not look at this text right now. Let me use a picture to explain, right? This is important. You may have heard about the sequencing of the human genome, right? The sequencing of the human genome took like about $4 billion and 13 years to complete, all right? That means figuring out each of those letters, all right, in a human genome, right? Take somebody's human cell. You want to find out the approximately 3 billion letters, okay, what they are, okay? And it was a very, very laborious procedure, right? And I'm going to present that to you right now. And you will see why it will take so much time, because you're going to sequence one base at a time. Okay, do that like 3 billion times, then you get the genome, right? So $4 billion, close to $4 billion in 13 years. Today you can do the entire genome in about 24 hours. Okay, you can do all of that. Why? Because this is because of parallel processing. Instead of trying to sequence this 3 billion of these guys, cut it up into pieces, right? About 100 units long, right? And every time it's a different cut, right? And then sequence those guys, because now you can do them in parallel. Okay, 100 units won't take that long to sequence. Do that many times and cut it several times. So this is something like, taking an encyclopedia, right? You want to read the whole thing. Okay? You don't have time to read one word at a time. Just cut it up. Just pass it through a shredder, okay? Every time you pass it through and maybe the shredder is doing one word at a time or something. Every time you pass it through the shredder, the cuts are random, okay? But then, so it's made the problem a lot easier, but then you have to piece that whole thing together, right? to get back to the original sequence, right? It's like a puzzle, you are going to have to solve that. So you've broken the problem down, right, to be able to, work on it a lot faster, but then you have to put the pieces of the puzzle back together, right? So that is called next generation sequencing, right, which I'm not going to cover in a whole lot of detail, but I mean, I don't have time to cover that in detail, but I'm telling you what the big picture is. Instead, what I'm going to cover here is what is called Sanger sequencing, right, which is how you sequence the DNA one base at a time, right? There also the underlying principles are the same. It's just that that you're using the technique of divide and conquer, right? You're using the technique of divide and conquer, and the method you use for what is called base calling, right? Calling the base. What is this base? Or you're making a base calling. That might be different. It might use an engineering approach where, when the DNA bases they pass through something, different voltage is induced, or there is a camera that's taking a picture. Lots of possibilities are there, and there are lots of people that are working on those things. Okay, but first let's try to understand the basics of Sanger sequencing. All right. Now, if you remember, all right, when you're looking at the formation of a DNA strand, all right, you basically have these uh, deoxy uh, ribonucleotides, okay, enter the reaction essentially as triphosphates, all right. So in order to form a new DNA strand, all right, you have a template. Looking at that template, all right, DNA polymerase is going to basically add new nucleotides, right, to the existing strand, right, in the 5' to 3' location. Okay, the mechanism, see, it's like this strand has got this 5' location, 5' end where you have these 3 phosphates, right, but it also has a 3' location on the hydroxyl group on the sugar, right. And remember, this is deoxyribose. So what do you have here at this location? If you had ribose, what would you have at this location? OH. If you have deoxyribose, you will have just H. Okay, remember that. Okay, so that is deoxyribose. Okay, the sugar is deoxyribose. But at this other location, at the 3' end, you have an OH. All right, and the way the chain elongates is because when the next nucleotide comes, okay, the phosphate at the 5' end of the next nucleotide is going to be linked to this hydroxyl at the 3' end through a what bond? Phospho diester bond, right? And this is an energetically unfavorable reaction. That's why you need these triphosphates, all right? So that this thing is cut out, and that supplies the energy for driving the reaction forward, right? That's how DNA is formed, right? Now, what happens if instead of this sugar here, that is deoxyribose, I have dideoxyribose? That means here also instead of that OH, I just have H, right? That means that chain is now dead. It cannot be elongated any further, because the phosphate of the next nucleotide cannot come in and latch on here because that OH is missing, right? It's become an H, right? So that's the main idea on which this thing is based, okay? So if you want, let's say, if you want to get different strands of DNA, let's say terminating at the A's, okay, what you could do is if you're doing a DNA synthesis reaction here, let's say the lower one is the template, okay, or this is the sequence, this is the strand that you want to sequence. Okay? You want to find out all these letters, okay? What you can do, so first of all, before you can do the sequencing, you will need some nucleotide, oligonucleotide primer. Okay, some information you need to have because you cannot start it from scratch. Because some matching strand you have to have here, or a little bit. Then you start the DNA synthesis reaction. So normally, if you didn't have this if you didn't have this dideoxyribonucleotide, what would happen is that the bases would come, and then you have a C here, so G is going to form here. Then you have a G here, C is going to form, and so on. Okay. So you will get the complementary strand, and it will stop, at the end. Now, if you also add a small amount of this dideoxyribonucleotide A, then, in some of the strands, let's say at this location there is a T, A got incorporated, all right? But the A that got incorporated may not be the deoxyribonucleotide. It might be the dideoxyribonucleotide. Then this strand cannot elongate any further, okay? So if you add the right amount, and again, I'm not a biologist, I don't know what exactly the right amount is. If you add the right amount of these dideoxyribonucleotides, right, then you will make sure that at least there will be one strand, right, that will terminate at each of these A's, okay, which cannot be elongated any further, okay. So this is for A, but you have 4 bases, right, A, G, C, and T. So I'm going to run 4 different DNA synthesis reactions, all right? In the first one, I'll add some of this deoxy A, all right? Deoxy ATP, dideoxy ATP, all right? Then in the second one, I'll add dideoxy, let's say GTP, then dideoxy TTP, right? And dideoxy CTP, right? So, any questions so far? So now look what is going to happen, right? So I'm having 4 different DNA synthesis reactions that are being run on the same strand, right? That you're trying to sequence. You have the primer, right? Oligo primer for starting the reaction. So if you're looking at the first lane, okay, the reaction here, all you've done is you had the deoxy ATP, deoxy TTP, deoxy CTP, deoxy GTP. This is the normal one, right? But then in the first lane, you have added some dideoxy ATP. As a result, what is going to happen is that, there's going to be some strand where this dideoxy thing got incorporated. That strand is not going to elongate anymore, all right? Similarly, at the second location, all right, again, there is a dideoxy A that got incorporated. This is not going to elongate anymore and so on. Okay, looking at the sequence at the bottom, the complementary sequence. So wherever there is a T on this first one, you're going to get an A, all right? And in some cases, the dideoxy A is going to get incorporated. Okay? Same thing with the second one, but except that here the focus is on the T's, the third one, the focus is on the C's, and then the fourth one, the focus is on the G's. Okay, you see that? Now, if you run gel electrophoresis, okay, which one is going to migrate the furthest? If you run gel electrophoresis on all these 4 lanes, let's say the voltages on all are identical, all right? The shortest strand, right, is going to migrate the furthest, okay? So the furthest migration you will see in the A lane, okay, on the left, right? The next one you're going to see in the T lane, right? And so on, right? Now if you start reading from the bottom, you'll get the sequence because it's A, T, and then whatever this is G and so on. So you're going to get the sequence, okay? So this is how the human genome was sequenced, right? And it took 13 years, all right? And $3.8 billion, all right? To find out the entire sequence of a human being. All right, so this is called Sanger sequencing, but as you can see, it is very, very labor intensive, right? And you'll have to do this like 3 billion times, right? Okay? I mean, because you need 3 billion letters, right? I mean, so, well, I mean, not 3 billion times, but at least 3 billion letters you have to sequence, right? But it's going to take a long time to do. So today's so this is Sanger sequencing, right? So let me just go through the text and you will see that whatever I wrote down here, everything makes sense, right? So the key idea behind this sequencing is that if during DNA synthesis a dideoxyribonucleoside is incorporated into the growing DNA strand instead of a deoxyribonucleoside triphosphate, then the 3' end of the DNA chain is chemically blocked and therefore the chain cannot elongate any further. Thus, if we add DATP, DCTP, DGTP, and DTP in excess, that's the normal ones, all right, and a small amount of the dideoxy one, then some, let's say ATP, then some DNA strands complementary to the given strand and terminating at the various A locations will be produced. And based on this, we outline the following procedure to determine the nucleotide sequence of a given DNA strand. And this is just text that explains what I already showed you on the diagram, right? Okay, and you can read through this. Okay, any questions? Now, by subjecting double-stranded DNA to high temperatures or a high pH, the 2 strands of DNA can be made to separate, right? And this is referred to as denaturation of the DNA. But by slowly cooling the DNA or lowering the pH, the DNA can be made to renature, that is, reform the double helix. The nucleic acid hybridization-based techniques make use of the complementary base pairing properties of the nucleotides. A always pairs with T, G always pairs with C. So you can always use the complementary strand as a probe, right, if you're looking for something. And one application of DNA hybridization is in the prenatal diagnosis of genetic diseases, for example, sickle cell anemia, right? And examining a single gene in the human genome requires searching through a total genome of over 3 billion nucleotides, which is quite challenging, right? if you're looking for it. But then if you use hybridization, right, you could come up with a probe for the normal globin gene and a probe for the mutated globin gene, right, and then carry out the detection. So I think we're running out of time. Let's stop here for today. I just want to stop with this picture. On the left you see a nice DNA double helix with all these hydrogen bonds, all right? So if you subject it to high temperature or pH, it will denature the DNA, right? It will become single-stranded. But then when you do slow cooling, again you get back to this double helix, right? But this hybridization of DNA, it is very specific, all right? So if you have the complementary strand, all right, something binds to the complementary strand and binds really well, then you know that you've found what you're looking for. The complementary strand can be used as a probe. So let's stop here for today, right? I'll continue from here next time.